Okay, let's begin with part two of lecture one. And the main topics covered here are cell theory, the development of cell theory, the idea that cells are the fundamental unit of life, and a little bit on cellular evolution, origin of life, and origin of the first self-replicating macromolecule. I'll always give you a list of reading associated with the particular lecture. Here you can see there's a couple of assignments from the Alberts et al. Text and focus mostly on the chapter 1 reading. Chapter 2 knowledge is something that is assumed. Most of that will come from your time in chemistry or in other courses. And then there are a couple of other non-text book readings that I've made available on Moodle. The first is a review article from Science. This is a review by Nobel laureate Paul Nurse, where he really does a great job of giving us the basic history of cell biology as a discipline. It's a really good read, something that you ought to check out. And I've also given you an article published in Cell, which is focused on some new and emerging roles for RNA. So that will form the contextual basis for today's lecture. Now let's move into the slides. I'll always begin with an outline to give you a sense of where we're going and what our main learning objectives are. First, I would expect you to have a working understanding of the development of cell theory and a little bit of information related to the history of cell biology as a discipline. We'll then again take a look at the origins of cells, what our best models tell us regarding the way that the first self-replicating macromolecule evolved. And then we'll take a look at the central importance of RNA in that evolution because of the ribozyme hypothesis and the RNA world idea. Remember that Friday, Dr. Shaven will be lecturing to us about macromolecules and modern cell molecular techniques. And I want to remind you that one of the overarching principles of this course is the empirical approach. And so we'll begin that discussion as early as Friday. And again, once we approach new material, we'll always try to do it within an experimental framework. So we'll start building that toolkit of techniques, model systems, and approaches that will help us to better dissect this complicated discipline that we're studying. Another quick reminder, it's always important to remember where we are in the story of the course. And if you think of this course, as we talked about before by way of metaphor, as a book, the book has 3 parts. We are in part 1. We're defining the cell and its study, and this would be considered chapter 1. Okay, so it's always helpful when approaching a complicated discipline to understand the historical underpinnings. And what I mean by this is if we can understand the way that the field evolved from its very beginning, we can see the questions that the scientists were struggling with, we can see the next logical steps, and we get better insight into the scientific process if we approach it from this historical perspective. So we'll often try to do that in this course. So thinking about the development of cell theory is one important sort of first step along that path. So let's talk about that. Cell theory really began in the early 1600s with the invention of really complicated lenses in Holland. And at first, these lenses were, by today's standards, fairly rudimentary, but they were able to magnify images 2 or 5-fold. Think of them as sort of like a complicated version of a magnifying glass that we would see now. The technology had advanced to the point that pretty good magnification could be achieved by the middle part of the 17th century. And Robert Hooke was able to get magnification of up to 30x. So pretty decent considering, we're dealing with 17th century lenses. Hooke was able to take sections of plant material, and typically he would work with the bark of trees. He would slice it very thin and mount it on glass, and then use this sort of microscope to investigate what he found. And these are producing, again, about 30x magnification. And he used these lenses and these rudimentary compound light microscopes to make these very beautiful drawings of the plant material that he saw. So here's one example. So looking at this, you can see the sort of what we now would consider, of course, cells, these little compartments that he saw lined up within the field of view. And he called those cella. That's Latin for small room or cubicle, and that's where we get the word cell. That's the etymology of that word. So with technological advance in Holland, then combining that with Robert Hooke's interest in plants and his drawings of plant material, we get this idea that the plant material is made up of these repeating units called cells. Schleiden and Schwann were the biologists who then took that idea and proposed that the cell would be the basic fundamental unit of life. Now we're a little bit further along. We're in the 19th century now when Schleiden and Schwann did that. Now, biology isn't existing in a vacuum. Physics was going really full steam ahead in the 19th century. And John Dalton, in particular, was in the midst of developing the atomic theory of matter, the idea that all matter is reducible to atoms. So Schleiden and Schwann were certainly influenced by John Dalton's thinking, and that helped them to propose the idea that the cell would be the basic unit of living matter. Kind of funny, though, what Schleiden and Schwann believed at the time was that all of this was akin to crystallization and rather spontaneous, and of course we know that that's not the case. We know now that cells beget cells, or cells come from cells is another way of thinking of that, and of course that's because of mitosis. A cell goes through M phase of the cell cycle, and then we go through prophase, metaphase, anaphase, telophase, and then cytokinesis produces the two daughters. So of course that's how cells are born. But in the 19th century, we can forgive them for thinking that perhaps it was more spontaneous generation. So if we're going to state the tenets of cell theory, there are two of them. First, cells are the fundamental unit of life, and usually we throw in this and basic unit of structure. So the concept here is cells make up living material, and they're the basic unit of structure and function. And then the other tenet is the idea that cells beget cells. And that one comes from a couple of Germans, Virchow and Remak. And this is what I was alluding to before with the M phase of the cell cycle. So mitosis is how this works. A couple of other important events along the way that are indicated here. Robert Brown was the person who first discovered organelles within eukaryotic cells. He discovered the nucleus in the early 19th century. And then Louis Pasteur, who we think of as one of the early microbiologists, was able to finally debunk the idea of spontaneous generation in 1860 with his swan neck flask experiment. And this one's kind of cool. You take a conventional Erlenmeyer and you heat up the tip, and then you kind of extend it out like this to make the so-called swan neck. And before you've done all that, you fill it up with a nutrient broth that microorganisms like to grow in. And you can apply heat, and you can boil this to sterilize it, and you can sit it on a bench top. And you can leave it there for a very, very long time, and nothing will ever grow in the culture medium. It will not become frothy or cloudy because nothing is able to gain access to the nutrient broth. So nothing can spontaneously arise in the nutrient broth. And to sort of drive home that point that you need some inoculation to get growth, you can break off the swan neck and now return it to a conventional Erlenmeyer and set that on the bench top for a week. And when you come back, you'll see it cloudy and frothy and full of microorganisms. That's because dust particles have settled in, carrying bacteria, and they've taken over that nutrient broth and grown like crazy. So Pasteur devised this very clever sort of experiment to show that spontaneous generation isn't possible. We need an inoculation. In other words, we need a cell to beget other cells. So the combination of these two ideas formulates the basis of cell theory, and we still think about that as the working principle of cell biology today. Over here, a couple of cool looking cells. Here's a bacterium, and here is a complicated neuron called a Purkinje cell from the cerebellum of a human. And you can see the complexity of cells, relative simplicity here with the bacterium, and pretty remarkable complexity here for the neuron. But both of these share the same biochemistry, the same genetic material, the same information flow. They utilize the same biochemical entities to drive their function. And really, that similarity is evidence of evolution. It's certainly evidence of cellular evolution. And I want to spend a couple of minutes here on this slide, and I'm going to read some of this to you because I think it's important, but I just want us to spend a moment thinking about this. We look out at the world, and we see remarkable organism diversity. You know, the plant, I'm looking out my window right now, and I'm looking at all of this complexity of plant life out there. I can even see a few rodents that are gathering, food. It looks very complex, it looks very different, and macroscopically there is remarkable diversity. But when we use a microscope, when we look inside the cells of the plant or the rodent or even the human, what we see is a lot of similarity. And that underlying similarity is there because of cellular evolution. We all started with a universal common ancestor. So it's not surprising that even though we're different at the macro level, at the micro level we're quite the same. It's very interesting. So here are some of the examples of similarity. All cells use DNA as the store of their genetic material, and this is what is replicated in S phase of the cell cycle to be passed on to the two daughters after cytokinesis. All cells use the same central dogma of information flow. Remember, the central dogma is this concept, DNA to RNA to protein. DNA can be replicated during S phase. DNA can be transcribed by RNA polymerase into RNA. And then RNA can be translated by ribosomes and rRNA and tRNA and etc. into protein. That's the central dogma. That is the same dogma. That is the same for all cells. Similarly, all cells have membranes to separate them biochemically and to protect them from their surroundings. All cells use ATP as their energy currency. We saw in the last part of this lecture how we can trace the high energy phosphate bonds in ATP all the way back to sunlight. Everything is connected. Whether you're a single-celled organism or a complex human, it's all the same. Here's a really good illustration of that idea, this is what we call substitution of gene products. So if you grow yeast long enough, and people have done this in the mid-20th century, you'll start to find mutant strains of yeast that stop going through the cell cycle and they arrest in the cell cycle. So we've known that since about the middle 20th century. Well, in the mid-70s, people started to figure out the genetic basis for this and started isolating some of the genes that are responsible for cell cycle and for the cell moving through, from M phase into G1 into S into G2. If you take a human gene that drives the cell cycle and you introduce it into a yeast, you can rescue the cell cycle defect. And what that means is that the human gene is similar enough to the yeast version of that gene that is no longer working, that the human gene can substitute for that mutated yeast gene. It's pretty amazing that you can do that. We see similar things in transgenic mice. You can make a transgenic mouse model of Alzheimer's disease by introducing a mutant form of human APP, for example. So people have been thinking about this idea of macro diversity, micro similarity for a long time. Lewis Thomas was a physician biologist, sort of autodidact who was into a lot of things. who in the early 1970s wrote a book called The Lives of a Cell. This is what he said on this topic. The uniformity of the Earth's life, more astonishing than its diversity, is accountable by the high probability that we derived originally from some single cell, fertilized in a bolt of lightning as the Earth cooled. It is from the progeny of this parent cell that we take our looks. We still share genes around, and the resemblance of the enzymes of grasses to those of whales is a family resemblance. Pretty cool. You can overlook the hyperbole, fertilized in a bolt of lightning. My gosh. Okay, so what is it that drives all this macrodiversity? Well, of course, it's the genetic change that occurs in evolution and provides us with the fodder for natural selection. And what we mean by that is, here's an idea of an original gene. Some random mutation occurs that may confer a selective reproductive advantage in offspring. And if so, then that mutation will be maintained in the population. That's the classic Mendelian idea of evolution via natural selection that Darwin first laid down without really knowing the chemical basis for it, of course, but all of these ideas do relate back to Darwin's thinking. Another way of thinking about this is here's the original gene. Now we have some crossing over event in meiosis that produces a gene duplication. This happens a lot in eukaryotes, and it's responsible for a lot of the different flavors or isoforms of proteins that we see in eukaryotes. It's why we have one version of yeast, actin, but 8 versions of actin in human cells because of gene duplication. Another example would be deletion, where for whatever reason, during probably a crossing over event, part of a chromosome is not brought over, or shuffling of exons, where we have sort of rearrangement of reading frames and rearrangement of coding exons to produce very different protein products. All of these are genetic change that can drive evolution by natural selection. All of these up here we call vertical types of genetic change because they're passed vertically from one generation to the next. Another way, and a much more dramatic and quick way to drive genetic change, is via horizontal transfer. We see this in viruses and in bacteria all the time. The endosymbiotic theory of mitochondria, for example, the idea that an early eukaryotic cell engulfed a primordial prokaryote, and now that primordial prokaryote is living as an organelle, mitochondrion, in the eukaryote. That idea is very much in line with this concept of horizontal transfer. So here we have, the genetic innovation is this particular red gene from organism A is transferred into organism B, and now that may provide organism B with a selective reproductive advantage that would allow it to be maintained in a population. So whichever mechanism it is, vertical or horizontal, this is what drives evolution. And it helps to explain the internal similarity despite the macrodiversity that I was alluding to before. So how can we study the origin of life here on Earth? I mean, we know from ice core sampling, we know from looking at radioactive isotope dating, relatively how old the Earth is, 4.5 billion years, whatever. But we can't obviously go back in time and observe the origin of life, nor do we have any record of it in the fossil record, because of course what is recorded in the fossil record is typically large bony structure. Obviously we didn't have anything that complex when life was evolving, 3.5 or whatever billion years ago here on Earth. So we try to simulate it in the lab. We try to do what is called in vitro evolution experiments, and there's a pretty proud history of that here in the Midwest. For example, at the University of Chicago, the famous Miller-Urey experiment was performed. This one is a classic. It's been replicated many times, and there's some examples of it still going on today. It's pretty simple by way of setup. You have, so this will remind you of your organic chemistry labs a little bit. You have this sort of glassware setup with a water source that you can heat to produce water vapor. And the water vapor will come in and then condense in a chamber that's filled with different gases. And what these gases are basically carbon dioxide, ammonia, methane, nitrogen, things that we knew were components of the Earth's early atmosphere. And again, we know that because of deposits in ice core samples and in soil and things like this, deep in the earth. So into this chamber, we can introduce a little spark of electricity. This is kind of like the spark that Lewis Thomas was talking about in the quote that I read earlier. And what you do is you then sort of condense it and cool it, and then collect the eluate, and then analyze it. And at first, of course, you don't have anything in there at all. But if you wait weeks, eventually you'll start to get simple organic compounds, including some of the simplest amino acids like alanine, for example. And what you'll get also are things like formic acid. Another product that we get in more of the modern Miller-Urey experiments is formaldehyde. And the reason this is interesting is because formaldehyde can spontaneously react to form ribose. And ribose is the pentose sugar that forms part of the structural basis for RNA monomers. And that's kind of interesting. And that is really an important observation in the formation of the RNA world hypothesis. The idea that in the primordial soup, in the early Earth, we get the formation of an RNA-based biology. Another reason that the RNA world hypothesis has gained so much attention, and it's really we've provided a lot of support for this idea, is because of what I alluded to at the end of part one of this lecture, the idea of RNA as a ribozyme or an enzyme. That kind of paradigm shifting idea that really broke down our previous conception of central dogma. The idea that RNA could do something more than just hold information. Not only could it hold information in the form of sequence, the sequence of nucleotides in a polymer, but it could also catalyze bond formation. Remember what I told you guys about the rRNA in the ribosome? rRNA in the ribosome that catalyzes peptide bond formation during protein synthesis. That's a really novel idea, but that's gained a lot of support over the years. We'll see lots of examples in our class of RNAs working as ribozymes. We'll see it with splicing. We'll see it with microRNAs. We'll see it with components of other components of the gene expression paradigm. So RNA is doing a lot to convince us that in the early Earth, in the primordial soup, RNA-based worlds were likely to evolve first. The Miller-Urey experiment is just one example of the evidence of that. So here's another look, or more formal look, at the RNA world hypothesis. This was developed in part by Francis Crick in the 1960s. Francis Crick, you will probably remember from Watson and Crick, their famous 1953 Nature paper that first described the structure of DNA. Of course, they borrowed the X-ray diffraction data of Rosalind Franklin and didn't give her credit for it, but that's a different story for a different day. Rosalind Franklin is one of the most important scientists in the development of the idea of DNA as the hereditary material. She doesn't often get the credit that she deserves. Yes, so RNA world, and what we need to be able to have for the RNA world to work is we need a macromolecule that can hold information. And what I mean by that is hold information in the form of sequence. So what you probably know about nucleic acids, like RNA, is that they are polymers of monomers. So a monomer is a single unit. I'm going to represent it as a circle. And if we have a bunch of these circles floating around and they carry information in the form of, hey, this one's an A, this one's a U, this one's a C, this one's a G, that's really not that useful. But if we can start linking those monomers together into what we call a polymer, well, now we have information. That information is in the form of the sequence. So AUG, CU, or whatever we make up. And we can define this as the 5' end of the molecule, and this is the 3' end of the molecule. There's information in that polymer in the form of sequence. And we need a molecule that can hold information in order for heredity to work. How else can we send the information from one generation to the next if we can't hold it in the form of sequence in a polymer? Now, RNA certainly is capable of doing that. The other thing that RNA is able to do that DNA isn't, by the way, is to act as a ribozyme, to have enzymatic activity. There are no known examples of DNA molecules acting as enzymes. There are many, many examples of RNA molecules acting as enzymes. So it's convenient to think of RNA as the early macromolecule of life because of these two properties. The fact that they can hold, RNA molecules can hold information in the form of a sequence in a polymer, and because of their ribozyme activity, they can do things like self-replicate. So if we imagine a simple self-replicating early macromolecule made up of RNA, it's not too difficult to imagine how that could be our universal common ancestor. And other in vitro evolution experiments have largely validated this idea. All 4 of the monomers of RNA have been difficult to produce in those in vitro evolution experiments, but give it time and maybe we'll see that happening. There has been the creation in an in vitro evolution experiment of an RNA ligase, which would be required, if you think about it, to link these guys together into a polymer. So there's proof of principle from in vitro evolution experiments from as recently as 2007 that that is the case. So when we think about the RNA world, we're thinking of about 3.5, sort of like between 4 billion years ago and sort of 3.8 to 3.5 billion years ago. That's usually what we think of. After that, we get DNA, and then we get protein and DNA sort of added into the mix. But it was all about RNA at the beginning. Here's an illustration of what I was trying to allude to before, or trying to indicate before. Here is a polymer of RNA, and that's what I mean by monomers being linked together. They're holding sequence, that's information that we could then pass on to the next generation, to the daughter. But that's not possible unless we can make a copy of that original sequence. So the idea is that an RNA-based polymer evolved in the primordial soup that was not only capable of holding information in the form of its sequence, but also of self-replication. So it serves that original sequence as a template for its own complementary sequence. And it basically builds a copy of itself. Now we get a double-stranded molecule where the molecules are sort of held together by the hydrogen bonds between the nitrogenous bases. You could then imagine these two breaking apart, and now you've got essentially the light blue version and the dark blue version. Well, if that dark blue version copies itself, guess what we have? Now we have the light blue version again. That light blue version, through a couple rounds of biochemistry, has essentially replicated itself, made a copy of itself. And now we can start to get changes in sequence that can lead to more complexity, etc., over billions of years of evolution. That's the concept. So the way we think it worked is in the primordial soup early on, RNA-based systems evolve. Eventually they form the ability to self-replicate. That's what that little looped arrow is showing. One will potentially evolve that can then build a protein from that RNA sequence. And then one could then sort of think of going backwards from RNA to DNA. And this is what we have in present-day cells. One of the reasons that DNA is so much more convenient than RNA is it's much more stable. RNA molecules are notoriously unstable, at least in cellular conditions in present day. So DNA is a much more faithful store of the information, if you will. Finally, it's one thing to have self-replicating RNA molecules in the primordial soup. I told you that RNA is very labile and very prone to breaking down. Wouldn't it be nice if we could somehow protect it from the vagaries of the environmental changes that might occur? Wouldn't it be wonderful if we could somehow encapsulate that early RNA molecule in a protective lipid bilayer? So enclosing the self-replicating polymer in the phospholipid vesicle could be very helpful, because now there's the ability to sort of make distinct the internal environment and keep it separate from the external environment. And the external environment may change, but the internal environment may be more stable and more consistent. And of course, what you know from other courses, I'm sure, about lipid bilayers is that it's a polymer of phospholipids. They line up like this. They self-arrange, by the way. It's kind of interesting. The monomers are amphipathic, so they have a polar head group and a hydrophobic tail. So the hydrophilic portion will face the aqueous environment of the extracellular space if we're talking about the outer leaflet, and then the inner leaflet would be here, and it would face the aqueous environment of the cytosol. We just line these up one after the other. So amphipathic, if that's not a word you're familiar with, amphipathic means containing both hydrophilic and hydrophobic domains. Phospholipids fit that definition quite nicely. And here we have the idea of a self-replicating RNA encapsulated by a vesicle of lipid. And conveniently, look at this. Simple fatty acids, those simple monomers that are amphipathic, are products of in vitro evolution experiments. So we have proof of concept that under conditions that mimic the early Earth, the sort of, pre-macromolecule phase of life on Earth, under those conditions, we can get the monomers, or at least components of them, like ribose, and we can get, not only that, we can get some of the phospholipids that would help to protect those early self-replicating macromolecules. So pretty cool. All right, that's the end of part 2 of lecture 1. And again, our main learning objectives here, we're trying to understand the development of cell theory, and then to understand how early life on Earth might have evolved. I would expect you to have a working concept of cell theory, what the two tenets are, and how what evidence we have to support those ideas. I would also expect you to be able to go from, the primordial soup to an enclosed self-replicating macromolecule, to be able to think about those steps. And remember, the way that we're going to look at whether you've mastered those learning objectives is by your completion of the discussion questions. And just a quick reminder, those are typically going to be posted on Friday and then due the following Wednesday. All that information is outlined on the syllabus. Take care, and Dr. Shaven will join us on Friday for the discussion of the macromolecules and for a look at some of the key model systems and experimental approaches in the discipline. Have a wonderful day. Take care.